Hello, my name is Zachary Schultz, and I'm going to tell you about our work involving nanostructures for biochemical detection today. So this work is going to demonstrate some microscopy techniques that are going to provide both chemical information of the sample, but also high spatial resolution imaging. All right, when we think about optical imaging, what we generally think about are these beautiful fluorescent pictures where we're not actually looking at the structure of the molecules, but rather dye molecules that have attached to different structures that give rise to the beautiful color. For example, here the yellow shown in the image on the right side of your screen comes from a stain that marks the nucleus. The magenta is marking mitochondria, and then the blue-green filaments are cytoskeleton markers. So preferred interactions between those molecules and those structures give us that color, but we're not seeing the whole picture around it. The idea is that the molecules that make up the cellular structures could also give us this information. So is there a way to do this without a molecular label or dye? Can we develop a label-free method of looking at biochemical molecules? All right, so the answer that we're going to turn to is something called Raman scattering. And Raman scattering involves using a laser to produce chemical information from molecules. When the laser is incident on a molecule, some of that light is scattered off as Rayleigh scattered light, which is the same frequency, but some of that light loses or gains energy and Raman scatters, either Stokes scattering at a lower wavelength or anti-Stokes scattering at a higher wavelength. If we measure all the scattered light coming off the molecules, what we see is that we can get a spectrum which then is representative of the molecule that's interacting. Using that spectrum, we can then identify the molecule. Now, the problem with Raman is that it's generally a weak effect, requiring higher concentrations in excess of 10 millimolar or more. And so we need a way to enhance that. So my lab has been using plasmonic nanoparticles as Raman probes. And so the idea here is that if that molecule is now near a nanoparticle where its plasmon resonance is excited, we can enhance the Raman scattering to the point that maybe we can see a single molecule. So this is what's known as surface enhanced Raman scattering. We consider this a probe because what we're looking at is using the nanoparticles to enhance the signal of something inside the cell that we haven't put on the particle to begin with. Think of it as the Voyager spacecraft. We've sent these satellites out into space, these space probes, as a way of returning information that we can't detect on Earth. So in our experiments, we're going to use nanoparticles as a way of going into cells to enhance the signal of biomolecules that we can't otherwise detect in the far field or from our normal instruments. All right. Tip-enhanced Raman scattering localizes a nanoparticle on the end of an atomic force microscope tip, an AFM tip. By moving that nanoparticle around in space, we can produce localized enhanced Raman scattering and generally create a chemical image. So shown here is an example of a nanoparticle tip, kind of an artist's rendition on the end of an atomic force microscope fiber, a cold glass fiber that we use. And you can move it around a cell. And so what's shown here is actually the topography of a rod photoreceptor cell. That same rod that's kind of shown down here in the image in the bottom left, where we have a long cell that's 10s, 50, 100 microns long, but we can then scan maybe a 2.5 micron image, produce a spectrum, a Raman spectrum, from the enhancement of the signal of that nanoparticle, and then plot how the change in that chemical concentration is varying. Shown here is an example of a TERS image taken and plotted for the intensity of 1655 wave numbers, which is generally representative of proteins. All right, the resolution now here is now related to the size of the nanostructure interacting with the cell rather than the diffraction limit of light, which would produce hundreds of nanometers resolution. And we, again, can get the Raman spectrum at each tip location. All right, one of the challenges in that last experiment was knowing where to scan. And this is where our nanoparticle probes have become useful. In this experiment that we published about a decade ago, we actually took a nanoparticle that had biotin on it that will then bind selectively to streptavidin. And so here we put streptavidin on a surface, bound our nanoparticle with biotin, and then brought our tip, our nanoparticle on the end of a TERS tip, to produce a very, very high Raman enhancement. It turns out that when you make junctions between two nanostructures, you induce a larger Raman scattering effect. And by doing this, we're actually able to produce a spectrum that is now representative of the protein on the surface that we were binding to. So now we have a way to probe protein ligand binding based on this nanoparticle interaction. And we can use the dark field microscopy and the AFM images to help locate where the nanoparticles are binding. All right, so that was kind of our proof of concept that this would work. Over the last decade, we've evolved this methodology from the stuff on the surfaces that I just talked about to actually looking at how these nanoparticle probes interact with cells. We did some work with antibodies on cells themselves, studying integrin receptors, showing that it's specific and selective. But more recently, we've moved away from the tips to using asymmetric nanostructures, nanostars. And I'd like to tell you a little bit more about how that works, because that adds some new advantage to these probes for chemical detection. All right, so nanostars, as shown in this TEM image in the upper left, have these branched points coming out. And if we look at this, what we have is kind of like a big center, kind of like our TERS tip, with protrusions coming off, which might be our nanoparticle probes. What we've done is we've taken this and then we functionalized it with a molecule that will bind to a protein of interest. In this case, the integrin proteins I referred to on the last slide, with the peptide arginine, glycine, aspartic acid, phenylalanine, cysteine. So RGDFC. Arginine, glycine, aspartic acid, phenylalanine, cysteine. The RGD motif has a specific interaction for the receptors, and phenylalanine, as the fourth amino acid in that structure, makes it selective for a specific integrin, in particular the alpha V beta 3 integrin. By making a cysteine the fifth amino acid in the ring, we can use gold sulfur chemistry to bind these to our gold nanostars. So we've done this, we've put this peptide on our nanostars, which now we can incubate with cells to try to detect and image the binding interaction in cells with that integrin protein. So using TERS, we were able to image about a 2 micron square region, similar to what I've shown you before. And again, we can use dark field microscopy to locate where those particles might be to help us target it. But now with the nanostars, the signal is intense enough that we can actually image entire cells and do 20 or 30 by 30 micron images of complete cells, seeing many more interactions to help understand the chemical information that's coming from our probes interacting with proteins inside the cell. One of the things we've been able to show is that this tests whether the binding is specific or not based on changes in the Raman spectrum that's detected. An example of this is shown here. This signature is what is detected from the RGD-FC peptide interacting with an integrin protein in situ, where all we have is for it to detect. And we can kind of show here the structure of this integrin protein we're interacting with. This is where the RGD peptide interacts with that. And by looking at the vibrations, the modes, the peaks that are seen in the spectrum, we can start to tease out information about how the protein is interacting with this probe molecule. In particular, we see a lot of vibrations associated with tryptophan, and we see there's tryptophan 129 here at the binding pocket, and then lysine 125, K125, also shows up in the Raman spectrum. And there's some other peaks in here that can be used to identify protein structure. Then by mapping the intensity of this signature onto our Raman image, we can tell where we're getting specific interactions from our nanostars interacting with our cells. Let's go back just one second. The challenge with doing this is that in this image now, we're diffraction limited again. So with our TERS imaging, we were able to look at things with 10, 20, 50 on a bad day in nanometer spatial resolution. In the wide field image, we're back to hundreds of nanometers. And we wanted to find a way to improve that. Turns out that we can apply super resolution imaging techniques to these nanostars interacting with our cells. So shown here is a cartoon where our nanostar has bound to a cell. And then we can look at this interaction on the cell membrane based on the observed Raman spectrum. A wide field image shown here in the middle shows that you see a lot of light. And what we've done here is, rather than mapping point by point, we're illuminating the whole cell and looking for changes in the scattering signal. By applying super resolution, we can then localize where those changes are coming from and regain the high spatial resolution imaging that we had earlier in our TERS experiments. So how does this work? Essentially, you have a signal that will blink in time. If we look at the frames, recording a wide field image over time. And if we fit the point spread function of the time fluctuating signals, we can do this throughout our movie and eventually localize where emission's coming from. And we see, for example, this spot right here, which corresponds to that brighter spot in the wide field frame that I've shown above, actually now has a reduced full width at half max of about 50 to 60 nanometers, as opposed to being diffraction limited at about 400 nanometers. And other people have developed this methodology. People like Catherine Willits and Nathan Lindquist have done some very nice work with Raman imaging. More recently, Alexander Brolo at the University of Victoria, showing that you can do super resolution SIRS imaging. All right, so here's just an example using that cell I was showing you kind of at the beginning where we have two cells together. And what I mean by frames is we're going to collect movies or frames. What I mean by movies, we're going to collect frames in time where at frame 1 we'll have one image. But if we watch this by taking, rapidly successive pictures, we can see here at frame 126, a new spot has appeared in our analysis. So in C here, what I'm showing you, or C in the middle here, I'm showing you kind of the average of all these signals. And if we look over here and look at how the signal in that box is changing with time, what we see is it goes up and down. And these spikes then become the points that we want to fit the point spread function from. By doing that, we produce this localized image that shows higher spatial resolution here in the bottom in D of this. And if we zoom back in on the box, we see structure now, whereas before it was kind of blurred out. Because this is Raman, we can still take the spectrum and identify peaks that we can correlate back to the signals that we're observing. And in this case, we see bands that are consistent with that tryptophan signal that we observed previously from our probes. All right. Using these probes, we can also use some other methods to analyze what's going on. So if we look at that localization, we see that it does localize to about a 60 nanometer spot. And shown here, if we blow that up, you can see the 60 nanometers spread out. But what's interesting is the color here now corresponds to the time at which the spot was acquired. And we see that there's a systematic movement of the spot over time. If we stretch that out in X and Y versus time, what we see is there's like a wagging back and forth. And what this suggests is that our nanostars may be altering orientation with the protein on the surface of the cell. If we look at a nanostar that doesn't have the peptide to bind to the protein, we don't see any of this wagging or improved localization. In fact, the super resolution algorithm comes back with something that's about 300 nanometers, which suggests that we're not super resolving it. It's moving around and producing a broader signal than is produced by a specific binding of these peptide nanostar interactions. All right, we can also use traditional particle tracking techniques and figure out the diffusion coefficient as shown here in the bottom right in E. And we see that the bare nanostar moves much quicker, consistent with the analysis I just told you about, than the one that binds selectively to the protein surface. So using this now, we're using this to study how nanoparticles are interacting with molecules as they move into cells. We want to follow the fate of these receptors. And some preliminary data in this direction is shown here, where what we've done is we've taken slices in different focal planes as our nanostars have interacted with the cell, so we can kind of get an idea of internalization and potentially different chemical actions that occur with that. Shown over here are the different slices, so at 0 micrometers, and then moving down into the cell, -1, -2, -3, -4, -5 micrometers, and where we've identified nanoparticles are shown by those squares. If we average that, that's the picture in blue over here on the right, and we can pull out those 4 different particles, and we can see that they're spatially distinct, suggesting that the nanoparticles really are being internalized, also showing you the zoom of those different particles around that image. So now we have a way to use this Raman signature to characterize the chemical interactions, but also resolve it in space, in volume through a cell. All right, to summarize what I've told you, we're looking at chemical-specific interactions of receptors in cells, and the functionalized nanoparticles can selectively let us detect and monitor protein receptors. Our SIR signal provides a chemical metric of the interaction for targeting studies, which we can then use to validate identification. And this methodology actually works in live cells. We start to understand where these signals are come from. It turns out that tryptophan species will provide a surface-enhanced resonance Raman scattering signal under the right conditions. And this SIRS intensity is sufficient for letting us do this super resolution experiment to look at both high resolution chemical imaging of nanostructures and teasing out the activity of biological molecules. All right, with that, I just want to acknowledge my group, in particular, a few people who've done some of the work I've mentioned here. The TERS imaging was work done by Hao Wang and Li-Fu Zhao. Hao was a grad student in my lab, and Li-Fu was a postdoc for many years with me. The more recent work, the super resolution work, was Carlos Lima de Albuquerque. Sean Sloan Dennison was the one who developed our Nanostar probes. And that work has been continued by some grad students in my lab, in particular Brian Scarpitti and Chelsea Zoltowski, Kinsey Bevins, and an undergrad who visited us, Victoria Save. Also, Amy Morrison has been working on this project as well. Some of our understanding has also been in collaboration with Patrick Curry. Funding for this work has been the NIH for the most part, with some help from NSF and Research Corp for Science Advances. And with that, I thank you, and I hope you enjoyed my presentation.